Okay, so we'll move on now to the next speaker, which is Rafael Bezerra Bezerra de Lira from University of Groningen in the Netherlands. So please, Rafael, you can now put your presentation on.
Muted. Yeah, I think you hear me now. Do you see the presentation?
Yes.
Great. First of all, I would like to thank you for the opportunity that you've given me to present my work here. I'm very glad that I have the chance to speak about what I've been doing the last few years. But at the same time, I feel sorry that we cannot meet all in person in the beautiful city of Foz do Iguaçu, which I have myself never visited before, but always wanted. So my talk today is about membrane fusion system, in which we are able to determine membrane fusion intermediates, kinetics, and efficiency. Back almost 2 years ago, in the title, I also said that we were able to measure membrane forces, and for that, I used optical tweezers. But 2 years ago, or like since the last 2 years, is a long time, and using optical tweezers, I was running into some inconsistencies, so I'm not presenting optical tweezers data here. But anyway, I hope this presentation will still be very interesting. So this work has been done throughout the last few years in a few different places. So the first one was at the Federal University of São Paulo with Professor Karin Risky, where I did my PhD. Later on, I moved to the Max Planck Institute of Colloids and Interfaces under the supervision of Dr. Rumi Dimova. And more recently, I joined the group of Professor Walter Roos here at the University of Groningen, where I speak. So coming to the technical parts, membrane fusion is a very important and interesting process that is related to many biological events, including egg fertilization, neurotransmission, cell area regulation, and so many other processes. And although membrane fusion is related to all these seemingly unrelated processes, fusion undergoes the same successive and common intermediate steps. So here, this is a representation of a neurotransmitter vesicle docking at the interface with a plasma membrane of the cell. Initially, you have docking that is mediated by proteins. Then you have membrane contact area here. The next step, you have the merging of the outer leaflets of the membrane, and at which stage you have mixing of the lipids, so it's lipid mixing. And eventually, there is a pore that is formed at the contact point that leads to the mixing of now the inner leaflet, as well as the mixing of the aqueous compartments, these two compartments separated before fusion. Not surprisingly, this process is very complex and complicated to study in cells. And so many scientists develop techniques and systems in which fusion was reconstituted in vitro. And in these in vitro systems, basically fusion is studied by mixing populations of liposomes, which are to fuse. In one of these liposomes, typically you have two types of fluorescent dyes forming a fret pair. So this system is under fret. And if these vesicles under fret mix with non-labeled vesicles, you have an increase. So you have a dilution of these dyes, and then you have an increase in the donor fluorescence. And this is the typical output you have when you perform these experiments in a cuvette, typically. However, this kind of data is difficult to analyze quantitatively. It is intrinsically the average out of many millions of fusion events. So what you see is actually the average, not every individual fusion event. This is also prone to artifacts because what you're actually measuring here is the dequenching of the donor dye, and dequenching is not always associated with membrane fusion. And also because it is based on dequenching, some fusion intermediates do not lead to dequenching, and therefore these assays are blind to some intermediates, including membrane docking, for example. So what is then the missing gap of this kind of assays? So the first one is it has been very challenging to detect some fusion intermediates, as well as the kinetics directly from your measurement. And secondly, fusion necessarily involves merging of at least two compartments, meaning that your final vesicle will be of a larger size. However, this is not typically reported. So although you measure changes in fluorescence, you don't measure an increase in vesicle size, although it's intrinsically related to membrane fusion. So for that, we try to fill in this gap by using giant unilamellar vesicles. I'm very glad that I don't have to introduce you in details about it, since Fernanda nicely introduced you to it. But basically, there are different ways of imaging GUVs. You can use phase contrast. So basically use phase contrast in order to check for membrane permeability. So if the membrane is intact or not. Or if you can label the membrane with a fluorescent dye or encapsulate a water-soluble dye inside a vesicle, you can observe membrane processes or anything that can happen inside a vesicle. So the first question that we ask is, can we then use GUVs to unravel the intricate details of membrane fusion? Well, hopefully we could. So in our fusion system, we had small fusogenic LUVs. They are red labeled with rhodamine. This is the fusogenic LUV composition. So you have a lot of DOTAP, which is a positively charged lipid. And then you have the OPE, which is a cone-shaped lipid. And for the GUVs, we had green labeled GUVs that are negatively charged because you have POPG. And these two dyes, they form a frat pair. So we adapt the typical frat fusion experiment to GUV experiments. And in our system, fusion presumably is mediated by membrane charges. And then potentially we would be able to detect these fusion intermediates by looking both at vesicle morphology. So if you had only docking but no fusion, you would be able to see these diffraction limited spots sitting on the GUV surface, but no change in the GUV color since there is no lipid mix involved. If you had hemifusion, you would still be able to see these docking points, but a small, in addition to it, a small change in GUV fluorescence. And conversely, if you had few fusion, you would be able to see a much stronger increase in the red fluorescence in the GUV, as well as GUV increase in area. At least this is what we had in mind when we designed the setup. So this is a typical result. This is a result from a paper that is published already 2 years ago. I'm not going into details because I want to show you more recent data. But basically, in this very nice video, at least I find it very nice, you have this GUV that is initially green labeled. It looks relatively tense because it's exposed to osmotic solutions. And when we add the LUVs, you can see that the GUV will become red because of frat, and simultaneously it will increase in area because lipids are being transferred from the LUVs to the GUVs. So this is a clear indication that in our system we have fusion, few fusion. As I said, this has been published 2 years ago. We very proudly say that it made it to the cover of this very traditional journal, the Biophysical Journal. We are very happy about it. But now I'm talking about more recent data. So the next question we asked ourselves is, can we measure the speed of fusion? So can we determine fusion kinetics in our fusion system? For that, we performed some controlled real-time fusion experiment in which we also added a content mixing marker. So in our system, as before, we had a green label GUV here, but now we come with a micropipette in which we micro-inject these LUVs that at the membrane are red labeled, but also these LUVs encapsulate a white fluorescent dye, so white in our microscopy images. And if we had fusion, this GUV will, first, the membrane will become red, like before. It will also increase in area, like before. But in addition to it, since we had a content fusion marker, the inside of the GUV will become fluorescent too. And this is what happens. So we micro-inject the LUVs with the help of micropipettes. We see that the GUV membrane becomes red due to fusion, and this happens simultaneously with content mixing. So the dye that is encapsulated in the LUVs is transferred to the GUV, and simultaneously the GUV increases in area due to fusion, and this area in our case is stored in the form of membrane buds, meaning that membrane fusion in this process is associated with changes in membrane spontaneous curvature. We made this video along with phase contrast videos as well, so we could see other kinds of information. So this is the same video, it's the same sequence, but now seen under phase contrast, you can see that the GUV starts fluctuating because of an increase in membrane area mediated by fusion. The area increase is accumulated in the form of membrane buds. And you can see that at later stages, the GUV becomes permeable, meaning that there are pores formed in this membrane. So in the beginning, this is more detailed snapshot of these figures. In the beginning, you have a GUV that becomes red labeled in the membrane, and this is associated with the content transfer into the GUV core. And then as more fusion takes place, both membrane and content fluorescence increase. At this stage, the membrane is intact. There are no pores formed in this membrane. So that tells us that the membrane can take a lot of fusion without becoming permeable. So it can sustain a lot of fusion. However, at a much later stage, when fusion becomes very significant and you have many more buds in the membrane, the vesicle loses its integrity, meaning that the GUVs cannot cope with so much fusion. Since we have performed calibrated frat curves in order to quantify more physical important parameters, but not only the changes in frat, so we use this frat calibration curve that I'm not showing here in order to calculate the number of individual LUVs. So these LUVs, they are added with the help of micropipettes that fused in real time with this very GUV here. And what we see here is we have very fast fusion. So if you look at this graph in this linear range, you have up to 5,000 single LUVs fusing to this very GUV. This is the fastest fusion system ever reported. And at saturation, you have nearly 10,000 single LUVs fused to this GUV. And we find it remarkable because no GUV, no membrane fusion system has ever reported such an efficient fusion process. And as I mentioned before, fusion is associated with spontaneous curvature. So the GUV membrane starts tubulating. We measure the diameter of these tubes. They become smaller over time, meaning that the spontaneous curvature increases over time. From the GUV bud size, we measure the spontaneous curvature in real time. And we see that the spontaneous curvature increases very largely until the buds formed are so small that we can no longer optically resolve them. So these experiments show us that we can not only measure lipid and content mixing and calculate the number of individual LUVs fusing to a single GUV, but we can also have mechanical information such as loss in GUV integrity, such as pore formation, as well as changes in membrane spontaneous curvature. And this is done in a single experiment, which is remarkable.
Well, 5 minutes.
The next question. Sorry, 5 minutes. Great. So all we've done so far was with the LUV composition that has 50% DOTAP, 50% DOPE. So the question we asked ourselves is, are we working with the most efficient composition? So our composition is made of DOTAP, which is a cylindrical shaped lipid, as well as the OPE, which is a cone-shaped lipid. And then we added a third component, which has neither of these properties. So it's neither charged, such as DOTAP, nor an inverted shaped lipid. So we made this fusion diagram. So this is just a representation. And then this is the three components we use, the OPC, DOTAP, and the OPE. And the composition that we've used so far was 50% DOTAP, DOPE, which falls in this diagram here. But instead of studying a single point composition, we wanted to map a whole diagram in order to obtain a composition spectrum. So we measure fusion efficiency by using FRET, but not intensity FRET anymore, but fluorescence lifetime imaging microscopy FRET. So when we don't have fusion, we have the longest lifetime. In our images, the GUVs will look red, like here. And the more fusion you have, the more towards the blue the GUVs will look like. So these are the typical results that we have. So for the compositions that we have studied so far, this is the lifetime map, whereas longer lifetimes mean no FRET, and short lifetimes, bluish, means more FRET. So for this composition that fuses very efficiently, you have high FRET. For the single component LUVs, when it's only DOPC or DOPE, you don't have FRET, meaning that you need charge. And when you have charged LUVs, you have more FRET, but not as high as when you have both DOTAP and DOPE. And these are the older compositions that we probed. We probed several different compositions, and then we built this FRET map of fusion efficiency. And basically, the darker colors indicate that we have more fusion. And the most efficient system we had here is the composition in which we had 50% DOPE and 50% DOTAP is this point here. That tells us that the most efficient system needs a lot of charge, but it also needs a cone-shaped lipid in order to destabilize the membrane upon contact. The last question that we are able to answer so far is, do these compositions that favor fusion fuse via few fusion? The answer is, For all the lipids for all the LUV compositions that efficiently fuse, fusion is mediated via few fusion. So we have content mixing. With that, I would like to summarize the work, saying that we developed a GUV fusion system that covers, that fills in the gap from previous fusion systems, that we are able to determine the composition of the GUVs after fusion, including the number of LUVs that fuse to the GUVs in real time. We determined the composition of the LUVs that most efficiently fuse to the GUVs. And we show that membrane fusion is necessarily associated with mechanical effects, including membrane permeabilization and changes in spontaneous curvature. With that, I would like to say thank you to all of you, to the organizers for putting us all together. And my dog says it's time for me to stop. Thank you all.
Thank you, Rafael. We have time for questions. Oh, there's one question here. Let's see. Rosangela?
Thank you, Rafael. Thank you very much for your very nice presentation and very exciting also work. It's really nice to see this work. And Rafael, I was wondering, because I was wondering at the same time I'm thinking about, because you said that you need charged lipids and conditioned lipids, because when you are explaining, I was thinking about if it is possible to have also fusion when you have rafts or lipid rafts. Have you tried this? Like if you have really the domains in the membrane, is it impossible to see fusion?
This is a good point. This is something that I'm working in collaboration with another student from Karinski's lab. And yeah, the answer is yes. So as long as you have charged membranes, you can have fusion with phase-separated GUVs. And fusion is most efficient when the membranes are more fluid. So if you have phase-separated membranes, you have one phase that is more fluid and another phase that is less fluid. And then you have fusion in both phases, but fusion is favored for more fluid membranes. But yes, the answer is yes, it's possible to have fusion.
But then you see more fusion in the liquid disorder phase or more fluid phase.
Yes, but the LUV composition is made of very fluid lipids. So in the end, we've seen this very interesting process in which we are transferring very fluid lipids to membranes, to GUV membranes that are phase separated. So in the end, we have so much fusion that we start dissolving these domains. And we can see that in real time very nicely. Okay. Yeah, so fusion is very efficient. It happens to phase separated membranes.
Okay, thank you very much. I wondered to see these results as well. Thank you.
Okay. There's another question here. Let me see who is asking. I could not find, but there's one question here. Oops. Okay. No.
I think there are no questions.
Yeah, Ana Cristina. Ana Cristina, can you say your question? Nope. Well, when we are waiting, Rafael, I would like to know, are you hearing me?
Yes, I am.
Okay. I would like to know, you said that this experiment of yours, the fusion is very probable because of the different charge of the LUV and the GUV. But what about if you trigger the fusion with, let's say, with calcium, would it be comparable? I mean, the amount of fusion you have?
We haven't tried that into much details, but in this system, when both membranes are charged, the LUVs and the GUVs, if we add calcium, it doesn't change much.
Yeah. I mean, I was thinking about not using the, let's say, the DOTAP, but using deuterionic lipids or.
No, what I know from experience is if you have GUVs of similar compositions, they are not charged, or even if they're charged, and if you have calcium, it induces membrane adhesion but not fusion. So at least from experience, it's not as efficient. Actually, it doesn't induce any fusion at all.
I see. That was my question. If you'd have transference of lipids, that's what I was wondering about.
No, we don't see that, but I didn't study that into details. So maybe it does in some specific conditions.
Yeah.
No, I think Andre, there's one. Andre Gomes wants to
Yes.
make a question. Andre, can you please?
Yes. Can you hear me?
Yep.
Yes.
Yes.
Okay. So, Rafael, congratulations for your presentation. I have actually two questions, if possible. One is very technical. You are watching a fast phenomenon of fusion. So, is there any specific condition for the lifetime images that you are acquiring? Since, of course, the quality of the data will be dependent on the number of photons you can acquire. So, I was if there was any specific condition. And another question is a follow-on on Rosangela's question. What would be the composition of the membranes you were seeing that have phases, phase separation, to study the fusion with these membranes, if it has cholesterol or only phospholipids that spontaneously segregate?
Yeah, very good point. So to answer the first question, in the flame images, we have this fluorescent dye that is very bright, is Body-Pi C16, and has a long lifetime. And we don't use a very high concentration, it's 0.5 mol percent, but it's very bright. So it's not a problem to observe or detect the fluorescence lifetime. However, we don't make movies, so we don't have kinetic information. So all these experiments are done under equilibrium after we incubate the GUVs and LUVs for 20 minutes. So yeah, we cannot have kinetics from fluorescence lifetime imaging, unfortunately.
Mm-hmm.
And the second question is the composition of phase-separated GUVs. These GUVs are phase-separated with POPG, not POPC, because we need charges, and cholesterol. And DPPG is the long-chain saturated lipid. So we have the short and long, we have the fluid and the less fluid lipids along with cholesterol. But both lipids are negatively charged, so we need charges in order for fusion to take place. So we replace DOPC or POPC with POPG and sphingomyelin or DPPC with DPPG. So we have more or less the same kind of membrane physically, but now they are charged.
Okay, thanks a lot.
Are there more questions?
Questions?
There's one question in the Q&A. Eduardo, can you see that?
Which one?
Oh, just
it's
small,
from
I can read it out loud.
Raya Sorkin. Yes, you can.
Oh, sorry. Yeah. Great talk, Rafa. Just a small comment. We do get fusion of zwitterionic membranes with calcium, but we do need a relatively high calcium concentration for that. The question is from a comment from Raya Sorkin.
Oh, thanks, Raya. Raya is a former member of the group where I work at the moment, so we know each other. Thanks for commenting on that. I will be curious to know a little bit more about this system. And also if you made more progress with the optical tweezer setup, since I didn't make much progress and I had to quit this project, unfortunately. But thank you.
Eduardo?
Yes.
I think we had a question that was sent to Dr. Prieto at the beginning that maybe we could go back to it.
Yes.
Okay.
So there's one question that was made at the beginning. I mean, it was made when we started the second talk. So there was a question from Emerson Carmona addressed to Manuel Prieto, and it's thanks for the talk. I'm wondering if you, in your FRET measurements in KCSA, you're also measuring solvent relaxation. Can you distinguish both processes? I hope Manuel is still there.
Yes. Okay. It could happen also that, and it's feasible. So, 2 points. First, the FRET data that we obtained, in fact, they are not affected by any kind of significant solvent relaxation, since tryptophan, it can certainly displace to the red, but not on this time scale. And also, it happens that they are close enough, so the FRET process is dominant. So that data is not biased by any kind of solvent relaxation contribution. We can think about a donor and one acceptor and determine its average emission spectra and calculate Forster radius and everything. Another studies that in fact can be done, it would be to introduce a solvatochromatic probe, lower than it would be a choice or some similar one into the channel, and just make a solvent relaxation study. Okay, it can be done and it's feasible. What happens is that in our study, one of the things that we really wanted, it was to use the native tryptophans instead of introducing any kind of exogenous probe that could certainly disrupt the structure of the channel or varying the distance by itself, etc. So that was one of the reasons why we did not went into that direction. But so the problem it would be to put that kind of probe in the selectivity filter, where everything is very fine-tuned and controlled. Using solvatochromatic probes in less critical regions of KSA, certainly, and well, it would be a valuable work. Thank you so much.
Mm-hmm.
I hope that I was clear.
I think it was. Are there any more questions? I would like just to point out, if there are no more questions, I would like to all the speakers to open their cameras, please. And so I'd like to ask everyone for a round of applause. And also tell you that our next webinar in 2 weeks will exceptionally be at 10 a.m. in Brasilia time.
Eduardo, is next week.
I'm sorry, next week. Exceptionally, 2 exceptions will be in 1 week, not in 2, and will be at 10 a.m. and will be transmitted in the YouTube link of the physics department of University of São Paulo, Ribeirão Preto. And the link is shown here. And you can also find it along with the full program of the Young Scientist Program series at the event website. So I think we're right on time. So we're sticking to the 2 hour that we have planned. So I'd like to thank again all the speakers for sharing with us such a great science and for the exciting talks that you have given us today. And we hope we can be together soon, presentially, not in this virtual format. And thank you again for your participation.